Good morning to all. Welcome to my YouTube channel, Mrs.MICROBIOLOGIST. So today we are going to discuss about a very important topic in microbiology, and as you all know, microbiology deals with various organisms. So let's go to the topic. So today we are going to discuss about staphylococci. You all know how important staphylococci is. So let's move on. So as an introduction, we can say we know all the bacteria we classify into two types, that is gram positive and gram negative. So this staphylococci comes under gram positive, and based on the shape it is a cocci. Cocci means a round shaped. So here you can see these cocci are arranged in grape like clusters. Okay, grape like clusters. So you know how grape like clusters are, they are round and it is arranged like grapes. Okay, grapes will be arranged like this, right? In a two-dimensional way we can say it is arranged like this. So this is how a staphylococcus can be seen. The next one is the most common cause of respiratory lesions in humans. So these wide varieties of organisms that staphylococci family includes, they cause so much of respiratory lesions in human beings, and it is a potential pathogen due to its ability to develop antibiotic resistance. Nowadays antibiotic resistance have so much of importance in our society, because day by day or majority of the organisms are getting antibiotic resistant, and it is very difficult to eliminate them from our body. Moving on, uh, this staphylococcus, okay, this word originated from the Greek words staphyle and coccus. So these are the two words derived from Greek word. These are Greek words. So let's see what is the meaning of it. Staphyle means a means bunch of grapes. Okay, bunch of grapes. Then the next word is coccus. Coccus means a grain or berries. Okay, berries are round, and you know grapes, how grapes are arranged. So then the pathogenic staphylococci produce golden yellow colonies. There are so much of staphylococci, pathogenic and non-pathogenic. So here we what they say is golden yellow colonies are produced by pathogenic ones, and white colonies are produced by non-pathogenic staphylococci. So moving on to the genus Staphylococci, as already we told that this is gram-positive cocci. These can in when we check the gram staining, it is arranged in singles, pairs, and irregularly as grape-like clusters. Not only grape-like clusters, it can be seen as pairs. Two cocci together, or single cocci can be seen in gram staining. So next, their characteristic is it is a facultative anaerobe. So that is a cultural character. When we culture, it can grow in not only in, it is not an aerobe. Aerobe means it only grows in presence of oxygen. It is a facultative anaerobe. That means it can grow in carbon dioxide containing medium also. So the very important biochemical character is this one, that is it is a catalyzed positive bacteria. That is a it is a very important character of that genus itself. All the staphylococcus are catalyzed positive, and it comprises around 40 species and its subspecies today. So moving on to the taxonomic classification, it comes under the Micrococciae family, and the genus Micrococcus and Staphylococcus. There are two genus in this family, Micrococcus and Staphylococcus. Then species, there is so much of species: Staphylococcus aureus, Staphylococcus citrus, Staphylococcus albus, Staphylococcus apophyticus, etc. So Then genus Staphylococci. First, we are this genus Staphylococcus is classified into two based on the coagulase test. Okay, based on coagulase test and its uh, reaction, it is divided into two. First one is coagulase positive, and second one is coagulase negative. So, in coagulase positive, Staphylococcus aureus is like Staphylococcus aureus was found out by Rosenberg in 1884, and other coagulase positive Staphylococci include Staphylococcus intermedius, Staphylococcus equus. So there will be always viva question relating to this, which are the like we know Staphylococcus aureus is a coagulase positive organism, but we don't know extra names which are other coagulase positive Staphylococcus. So this is a very important viva question. Keep in mind. Moving on. Then we are telling about the Staphylococcus. Uh, Negative coagulase negative staphylococci. It includes a variety of organisms, Staphylococcus albus, or it is otherwise called as Staphylococcus epidermidis, which is a normal flora seen in our skin. Then Staphylococcus citrus, then Staphylococcus saprophyticus, which is a very important coagulase negative. It is a common normal flora, but it causes honeymoon UTI. Okay, it causes UTI, urinary tract infections in or sexually matured or reproductive age group ladies. So Staphylococcus hominis, Staphylococcus capitis, Staphylococcus hemolyticus. So these are all coagulase negative staphylococci. Then moving on, we are going to tell about the structure and physiology of staphylococci. So here we can see it is a gram positive, as we already told, positive cocci with a grape-like cluster of pattern. Why these grape-like clusters formed? This is your question. It is because consider these are the round staphylococci, and here what happens is the cell divide along the different planes and the daughter cell remain attached. So these are two staphylococci, and its division takes place like this. Okay, different planes. Here what is the result will be is like this. So what happens is they attach, they don't detach. Okay, after the division, what happens is it doesn't detach. One cell divided into four. Here four, one, two, three, four, four, and it will be like this. Okay, it doesn't detach. The next cell also comes like this. So it combines and forms a grape-like clusters. I hope this is clear. And already this is a non-motile one. Okay, staphylococci are non-motile, so it doesn't possess any flagella or anything like that. Already told it is a facultative anaerobe, and it is a very important point that it is a salt tolerant. It is salt tolerant and desiccation tolerant, that is survival in formities. It will survive in like feces or vomit particles, etc. It is salt tolerant. Salt tolerant makes it grow in salt containing medias. Moving on to the morphology, we already told it is spherical cells, mostly arranged in irregular singles, pairs. It arranges grape-like clusters. The polysaccharide capsule rarely found on cells. So young cultures containing not all staphylococci, but some staphylococci containing polysaccharide capsule. Then it has a peptidoglycan layer, which is the major structural component of the cell wall, and the egoic acid is also present. These are very important. Okay, peptidoglycan layer. It is a gram positive, and gram positive containing major amount of peptidoglycan. Then morphology, continuing the morphology, as here you can see this staphylococci containing protein A is a major protein component of the cell wall. This protein A is seen in which one? Seen in the cell wall. Okay, this is protein A seen in the cell wall of staphylococci, and it is very important in escaping from opsonization. Here opsonization is a our defense mechanism. Okay, human beings defense mechanism to eliminate these types of bacteria. So what here, protein A is present in the cell wall of staphylococci, and this protein A helps in escaping from the immune system or opsonization. Okay, so next point is released into the culture medium during cell growth. Is how we can found there if there is protein A present or not. It is by when we culture this staphylococci, the protein A is released into the cell growth. So the unique property of cell protein A is the ability to bind to the Fc part of the IgG, not an antigen antibody specific reaction. So here what happens is this staphylococci attaches to the Fc part of an IgG3. IgG3 means it is an antibody. Even though this protein A is an antigen and IgG is an antibody, even though they combine, but this is not a specific reaction. It is a pseudo reaction. Okay, we will detailedly study about protein A in the coming slide. So next is cytoplasmic membrane. So we already told about the cell membrane. This one is the cytoplasmic membrane. It is made up of complex proteins, lipids, small amount of carbohydrates, serves as an osmotic barrier of the cell. It provides adherents for the cellular biosynthetic and respiratory enzyme. That is, this word means that it helps in attachment of the cellular biosynthetic and respiratory enzyme. Which one? Cytoplasmic membrane. Moving on, now we are telling about the morphology of staphylococcus aureus. uh, Up to this we told the general features of staphylococcus. This one is about the staphylococcus aureus, and the cocci is 1 micrometer in diameter, arranged in grape-like clusters. Cluster formation is due to the cell division in 3 plane. We already told with the daughter cells. Cells remains in close proximity. Then next one is that it is non-motile, non-sporing. Mostly small amount of capsular material is seen in non-capsulated bacteria. Even though it is a non-capsulated bacteria, some strains possess some capsular material seen in its above its cell wall. So moving on, next we are telling about the cultural characters. It grow readily on ordinary media, and its optimum temperature is 37 degrees Celsius. That is our human being's body temperature. Then it has a pH of 7.4 to 7.6, which is the optimum pH. In that pH it grows very well, and it is an aerobic and facultative anaerobe. That means it can grow in oxygen containing media, as well as in not carbohydrate, carbon dioxide containing media. Then colony morphology, if we tell about the colony morphology, the colony will be 2 to 4 mm in size, circular, convex, smooth, opaque, and easily emulsifiable. Easily emulsifiable means if we it is an opaque, okay, opaque media. If we take the plate and observe, we cannot see what is below that colony. Okay, it is easily emulsifiable. That means if we take a drop of distilled water and the mixed colony, what happens is it easily mixes with the water. So moving on, most strain produce yellow colonies on nutrient agar, and some also produce white, orange and yellow pigments. So here what happens is, we already told pathogenic ones produce yellow colonies, and non-pathogenic ones like albus produce white. Albus produce white colonies, that is epidermis, and yellow colonies are produced by citrus, etc. So then cultural characteristics, these staphylococcus colonies can produce pigmentation, which can be enhanced by using, we can use pigmentation, study pigmentation using 1% glycerol monoacetate or milk. We can enhance the pigment production by adding 1% glycerol monoacetate or milk. Then pigmentation, about the pigmentation, lipoprotein allied to keratin. Okay, the pigment of staphylococcus aureus is a lipoprotein allied to keratin. Then in nutrient agar slope, confluent growth or oil paint appearance, that is a specific appearance that is uh, only applicable to staphylococcus aureus. That is it has a confluent growth, means it has an abundant growth and oil paint appearance. The next one is blood agar. In blood agar, it can produce some strains of staphylococcus aureus produce hemolytic colonies, and others produce non-hemolytic colonies. Sorry for the background sound. My budgigas are so naughty, they are making sound. Okay, so the cultural characteristics, most strains incubated in 20 to 25 percentage of carbon dioxide. We already told that is a facultative anaerobe, so it produce hemolytic colonies. Marked hemolysis is seen in rabbit or sheep blood. Minimum hemolysis is seen in horse blood agar. In horse blood, ready-made horse blood agar, minimum hemolysis. But in rabbit or sheep blood agar, marked means very good hemolysis can be seen. In McConkey agar, small lactose fermenting pink colonies can be seen. So McConkey agar, what we can see is, this is lactose fermenting, so pink colored colonies, which means the staphylococcus are lactose fermenting. Then in liquid media, that the liquid broth, peptone water or glucose broth, etc., it forms a uniform turbidity. Then what are the selective medias that we can use for what staphylococcus? First one is Rutland's media, second one is lithium chloride telluride media. Then next is mannitol salt agar. We already told it is a salt tolerant organism. So milk salt agar or broth containing 8 to 10 percentage, can tolerate in 8 to 10 percentage NaCl. Then agar containing polymyxin. Okay, it can grow in agar containing polymyxin. For primary isolation, we use sheep blood agar plate. Okay, is BAP. Okay, sheep blood agar is used for the primary isolation. Then coming to biochemical reaction, I'm roughly saying all the main points of staphylococcus. We can in my profile, you in the playlist you can find specifically about different biochemical test reactions, including indole test, urease, MRVP and catalase, and many more different types of biochemical reaction. So here staphylococcus aureus is indole negative, then urea is positive, then MR test or methyl red test is positive, Vogus Prosker or VP test is positive, catalase is positive. Catalase, we already told, it is a very important test, because this helps in differentiating between Staphylococcus genus and Streptococcus genus, because all the Staphylococcus genus is catalase positive, in all Streptococcus, not all, majority of the Streptococcus genus is catalase negative. So in modified oxidase test, it is negative, but it is positive for the micrococci. Okay, micrococci. Biochemical reactions. Urease test is positive, gelatin liquefaction positive, lipolytic uh, properties it has. So it is dense opacity on agar can be seen. Then phosphatase test, which is positive because phenolphthalein diphosphate on nutrient agar exposed to ammonia. Sorry, this is ammonia. There is a spelling mistake. Ammonia vapor turn pink color due to phenolphthalein. Okay, due to the presence of phenolphthalein, what happens is when it is exposed to ammonia vapor, the it turn pink color. Okay. Then Staphylococcus aureus, which is coagulase positive. Staphylococcus aureus we classified into two staphylococci, coagulase positive and coagulase negative. So aureus is just coagulase positive, so it is very important, and it can ferment mannitol, and clear hemolysis can be seen on blood agar. It produces golden yellow pigment, and it liquefy gelatin, and it produce phosphatase and potassium tellurate medium. It forms black colonies. It produces thermostable nucleases also. Then penicillin resistance is a very important topic about staphylococci. It causes so much of resistance to antibiotics. First one is beta lactamase production, penicillinase A, B, C, D. Okay, so beta lactamase production means the staphylococcus produce an enzyme called beta lactamase, which blocks penicillin, which causes penicillin resistance. So this topic we have to describe in detail, so in the coming videos it will be uploaded. Change in bacterial surface receptors binding to the beta lactam antibodies. Changes in bacterial surface receptors binding to beta lactam antibodies develop tolerance to penicillin. So every time it bonds to the beta lactam antibodies, what happens is it changes its bacterial surface. Okay, changes its bacterial surface receptors. That is the main point. So in a bacteria, in a staphylococci, okay, consider this is staphylococci, there are so much of receptors present. Okay, so much of different types of receptors. So one is a beta lactamase. Okay, consider this one is beta lactamase receptors. So what happens is when penicillin comes and attaches to this, next time what happens is they changes this receptors. If the receptor is susceptible, then only it can bind to this receptor. So what happens is this organism, help with the help of a nucleus, what they do is they changes the receptor. Okay, changes the receptor results and escape from the binding of penicillin antibiotics, and hence it develop tolerance to penicillin. Then pathogenicity and virulence. There is so much of pathogenic and virulence factors present in a staphylococci. So we are telling the basic things, and the coming videos will containing elaborated and explained things. So cell associated polymers is one of the reason for its high pathogenicity and virulence. So it contains polysaccharide peptidoglycan, that is peptidoglycan seen in the cell wall. Then teichoic acid is present and capsular polysaccharide is present. Then we are telling about the peptidoglycan. Half of the cell wall weight is because of the peptidoglycan. As you know, this is a gram positive organism. This is a gram positive organism. So subunits of a peptidoglycan is N-acetyl-muramic acid NAM and N-acetyl glucosamine NAG. So cell wall is a very important component of a cell. So we have to study the detailed structure of cell wall. There is a video in my channel, you can go and check it out. Then unlike the gram negative cell wall, gram positive cell wall has many cross-linking bridge or layers, which makes the cell more rigid and rigid, causing a rigid cell wall. So here you can see gram positive has a strong cell wall, which it helps in escaping all the adverse environments. So let's see. There is teichoic acid present. The species specific phosphate containing different polymers are present, bond covalently. So how the teichoic acid is uh, bonded means it bonded covalently to the peptidoglycan. It bonded covalently to the peptidoglycan layer through the lipophilic linkage. Okay, this is the bonding, that is lipophilic linkage. Lipothioic acid is there. Then it mediates attachment of to the mucosal surface through its specific binding to fibronectin. Okay, now our body cells contain fibronectin. So what they will do is this lipothioic acid helps the organism to get attached to the mucosal surface of our cells. Okay, our throat containing mucosal surfaces and all that things. So it helps in attaching the organism to the mucosal surface through specific binding to fibronectin. Then capsule or polysaccharide or slime layers. So these are the things that is commonly believed to be in vivo, but it occasionally found when cultured in vitro. In vitro means outside our body. Okay, in culture media or in experimental animals, etc. So what happens here is this polycapsule or polysaccharide or slime layer. Slime layer is a semi solid layer which is seen outside the cell wall, and capsule is a strong rigid covering seen in above the cell wall. Okay, so it is found in vivo, means if the organism enters a body of a patient, and inside there they will produce capsule because they want to escape from all the our immune system's mechanisms, right? 7 cellular serotypes are identified in Staphylococcus aureus. So 7 types. Okay, uh, sorry, 11. Okay, it's my mistake. 11 types of uh, serotypes of capsular materials are present in different types of Staphylococcus aureus. In that, this fifth one and seventh one are the most important contributors to infection. This you have to always remember. Fifth one and seventh one account for major infection portion. So about the inhibiting, what are the functions of this capsule and slime layer is that it protects the organism by inhibiting chemotaxis, which is our defense mechanism. Then phagocytis, that is the engulfment of the foreign material and eliminating it from our body. So it helps this capsule and polysaccharide slime layer helps the organism from escaping from chemotaxis and phagocytosis, and by polynuclear leukocytes, or otherwise called as neutrophils. Okay, polymorphonuclear leukocytes or neutrophils. Okay, neutrophils also inhibit proliferation of mononuclear cells. Okay, what happens is the capsule will inhibit the mononuclear cell proliferation, because if one organism is there in our body, what happens is our cells, leukocytes will get activated and produce many more neutrophils. So this proliferation is prohibited by the capsule and polysaccharide layers. Then facilitate the adherence of bacteria to the catheters and synthetic materials. Synthetic, this is materials, that is a spelling mistake, sorry. Catheters and other synthetic materials. So this capsule and slime layer helps in adherence to many different plastic constituents. Okay, so here is about protein A, the surface of Staphylococcus aureus, but not cons. Okay, this is specific. Protein A is specific to which one? Staphylococcus aureus only. In cons means cons, the full form is coagulase negative staphylococcus. So the staphylococcus aureus is specifically coated with protein A and covalently linked to this. Protein A is covalently linked to the peptidoglycan. It has a unique affinity to Fc receptor of immunoglobulin IgG3. Actually, specifically IgG3, the protein A detection is one of the specific tests to detect Staphylococcus aureus. So it doesn't produce any specific reaction, but this protein A helps in detection. Okay, specific test for detecting Staphylococcus aureus. This protein A coated staphylococci is used as a non-specific carrier of antibodies directed against other antigens like streptococci. That is co-agglutination test. The test we use is co-agglutination test, because staphylococcus uh, coated with protein A. Staphylococcus coated with so much of protein A is combined with the serum. Okay, we take serum sample and it contain antibodies specifically to staphylococcus. If a person is having staphylococcus infection, then what happens? Our body will produce antibodies against staphylococci. If we have streptococcus infection, our body produces antibodies against streptococci, and these two antibodies will be different. So what happens here is this will command, this antibodies will come and attach to the protein A. If we have staphylococcus aureus infection, these antibodies come and attach with it and produce a agglutination. Okay, produce agglutinants which will precipitate in the solution. Okay, moving on, coagulase and other surface protein. Coagulase reacting factor CRF in plasma. Clumping factor is a bond coagulase seen on the outer surface. Okay, so here what happens is coagulase and other surface protein. Coagulase reacting factor is there in the plasma. In the plasma of our body, if there is infection, coagulase reacting factor will be there, and in the staphylococcus surface itself there is a clumping factor or bond coagulase. This coagulase reacting factor is also called as free coagulase. Okay, also called as what? Free coagulase. Free coagulase, that is not bonded to any surface, it is freely seen in the organism. Free coagulase. Then clumping factor is bonded to the outer surface. What these coagulases do is they binds with the fibrinogen and convert the insoluble fibrin. Okay, converts it into the insoluble fibrin. Okay, it binds with the fibrinogen and it converts the fibrinogen to an insoluble fibrin. That means it forms a clot. Okay, fibrinogen is a clotting element, and if we these plasma coagulase proteins will, what they will do is they binds with it, and okay, so clumping or aggregates will be formed. That means, when we determine the blood antigens. Okay, blood grouping, there will be clumps, right? There is a clotting seen in the test tube. And primary test, it is a very primary test for identification of staphylococcus aureus. Coagulase test is the primary test in identifying staphylococcus aureus. Other proteins include collagen binding protein, elastin binding protein, fibronectin binding protein. So okay guys, today this much is there. So thank you for watching. Like, if you like the video, like, share to your friends, and subscribe to my channel for more videos. And one more thing is that if you have any doubts, okay, any doubts, or if you need any class on specific topics, do comment in the comment section and let me know. If you have any feedbacks, you want to slow down, or you need to include more pictures or anything like that, the picture videos is coming. Okay, so if you have any suggestion, please leave in the comment section. I'm eagerly waiting for that. So subscribe my channel and keep supporting. So take care. Have a nice day.